These are really exciting new solar cells. Canadian researchers have used bacteria to produce a dye that absorbs sunlight. The great thing about the idea is that these cells could also work efficiently in regions with little or less sunlight. Is this a revolution of energy supply? Clixoom Science Fiction. Watch fascinating science videos thrice a week right here. Subscribe to the channel. Only then will you not miss a thing. A warm welcome.
1, 2, 3.
The research team from Canada used the bacterium Escherichia coli, E.Coli, for its experiment. We know it because it occurs in the human and animal intestines and can also trigger diarrhea and intestinal inflammation. The scientists have genetically engineered E. coli bacteria to produce large amounts of the dye lycopene. It ensures that light is effectively captured in plants. It is essential for the cells that carry out photosynthesis. For example, the dye is found in tomatoes and rose hips and gives them their red color. The scientists then coated the genetically modified bacteria with titanium dioxide, TiO2, a semiconducting material, and applied it to a glass substrate. And there you go, a new form of the solar cell was created. This actually generated a current density of 0.686 milliampere per square centimeter. According to the Canadian researchers, this is the highest current density ever achieved with such biogenic solar cells. In fact, twice as much as others have developed so far. In addition, all required materials can be produced sustainably and cheaply. Another advantage of this idea is that the dye does not have to be extracted from the bacteria, but the E. coli can be built in at the same time. However, according to the scientists involved, the main advantage of E. coli is its efficiency. No matter whether the sun shines a lot or a little, biogenic solar cells work just as efficiently with less light as with a lot of light. Project manager Vikramaditya Yadav comments, "We have thus created a basis for making solar energy more economical, cheaper and simpler. The problem is that the bacteria die. Therefore, in the long term, solar cells are to be developed in which the bacteria do not die but continue to produce lycopene." However, the principle of such solar cells in which organic substances convert the sun into energy is not completely new. A US research team had already built the first prototype of a biosolar panel in 2016. Specifically, 9 biosolar cells were combined into one solar panel. It was based on cyanobacteria that are capable of photosynthesis and survive within the experiment. This was achieved by using a carbon anode that had to be immersed in the liquid containing the bacteria. In this way, the air supply of the bacteria was guaranteed and the cell was able to work independently. The electricity was obtained from both the respiratory activity and the photosynthesis of the bacteria. A permanent power of 5.59 microwatts could thus be generated under the influence of light. Even if this doesn't sound like much at first, it could be used to power a wristwatch. However, such biosolar cells could be used in remote regions where regular battery changes are difficult and the connection to the power grid is not possible. But even if the research teams involved are optimistic, the metabolic processes of bacteria have not yet been sufficiently researched and, due to the rather low energy efficiency for practical applications, have not really been developed far enough to proceed with mass production. The further development of such ideas is very important in order to maximize the sustainability of power generation. And because this technology is still in its infancy, major leaps in the amount of electricity produced can be expected in the near future. Significant developments are therefore to be expected. Click here to find out how solar energy could actually become dangerous for us. Until then, stay tuned and see you soon. And click here.